Donna, I want to ask you, and Omkar touched on it here too. What are the operations steps for reducing batch-to-batch variability in manufacturing?
From my perspective and the intent of having a very consistent product at the end, there's a couple of things that are in consideration. As stressed a while ago, the importance of understanding if you're dealing with blood products, i.e. whole blood or leukapheresis, apheresis product, it's understanding that composition and understanding what may or may not interfere in your culture process. A lot of that can be managed in different ways. I use the example of red blood cells that are immature. Most culture systems, they will continue to grow unless you remove them. So I know a lot of early on companies will take a leukapheresis and take it straight into manufacturing, and or even if they select for their target seed material, it can interfere with that selection process from pure steric hindrance, if nothing else. So again, it goes back to automation, bringing in instrumentation that can effectively clean up your incoming seed material. If it's red blood cells, it's easy to use GMP DNA lysis solution that will, I'm sorry, not DNA lysis solution, a red blood cell lysis solution that will lyse those immature reds, and they won't come through into the clean process. We happen to use a LOVO. Our leuks come in and are automatically processed with a LOVO. It gives a much cleaner product. It removes all the interfering immature platelets that are often in our subject material. And it gives us a cleaner product to then go into our selection process and minimize the interference of those, quote, contaminants in our selection process. As you go through the manufacturing process and as you learn variability that does occur, you can start figuring out ways to address those variabilities. One of the things that I find helpful is to establish what we call an internal process control cell bank of incoming PBMCs, incoming selected seed material that are on healthy donors across repeated collections, that you establish how those healthy donors react across a group of healthy donors and within repeat collections of those healthy donors. And you do a lot of manufacturing processing through utilization of those donors that will let you see what to expect with the best of donors. And then when you get subject material that is varied, yields a variant, you can address how to best vary that. That's also helpful when you're trying to do tech transfer of your process to, say, a CDMO, CMO, being able to have that seed source material that you know the expected results with healthy donors that you can tech transfer across and make sure at least as you transfer to a CDMO that they get the basic process and get the results that you expect. Some of this comes with a lot more manufacturing, understanding your particular patient population. Again, what I've seen with hematological malignancies versus solid tumors is very different patient profiles. So what we do with the heme malignancies is a little bit different than what we do with solid tumors. So that's, again, a lot of it goes to training of the employees to understand those variations and what you can do to mitigate those. It changes along the way. One of the things that I think is important is to incorporate in-process analytical assays that give you some guidance. Something as simple as straightforward as cell counts along the way, population doublings, monitoring certain metabolites can yield different growth expectations. So you may have to feed a culture a little bit sooner if you've got a rapid grower than a more traditional grower, or in most cases with some of the, at least in our solid tumor population, we see that we definitely get a different growth curves than we do with the heme patients. And we make adjustments in the feed schedule and we do have automation. We automate it right out the gate from phase ones. So with that automation process, we have the means to adjust based on what we learn from the growth curves in the metabolites. So those in-process culture monitoring are important, and understand what you can do if you see variability in that.
Excellent. Thank you, Donna. That was an extremely detailed response, and I really appreciate all the detail, like I said. Omkar, before we move on to the next question here, I want to make sure that you have your opportunity to respond to. Again, we're talking about auto and the operation steps for reducing batch-to-batch variability in manufacturing.
Yeah, absolutely. And again, I love the detail in which Donna actually laid this out. But if I were to take a step back and look at it from an operations standpoint, as I mentioned earlier, look at the end-to-end journey, identifying those pressure points or failure modes or failure points. We had actually done a detailed FMEA analysis of failure modes and effects analysis to actually treat the entire journey as unit operations. So break it out, hone in on the points that actually are introducing the most degree of variability. It could be your raw material, it could be your media, it could be your lentiviral vector. It could be any parts. If you were to break it down into unit operations and address it, that's I'd say slightly lengthier, maybe a slightly more cost heavy development process as well. But that is the one way we've actually been able to go very, very detailed. And I'll give you a very specific example. One of my colleagues, Jennifer Rabin, who leads our industry working group, has been at the back. So she's an oncology nurse. And what she's been trying to do is, how do you reduce variability at the collection of the starting material for CAR-T, so apheresis, right? We all know that apheresis is mostly done at accredited centers. And what we're trying to push for is trying to do an 80-20 approach where an apheresis center is able to leverage much of its existing standard of care procedures, and the manufacturers can have an harmonized approach where, again, 80%, as I said, you're leveraging existing procedures, and 20% you're allowing for nuances in the protocol based on the manufacturer, right? So what you've done now is you've taken unit operations, and I believe an expert in the field and a dear friend, John Lee, in one of your webinars, he actually also mentioned something similar, where he was suggesting piecemealing the journey, identify the unit operations, and identify potential ways of standardization, right? So you have broken it down into smaller pieces. It's like attacking a bigger problem one step at a time. That's how, for an operations step level, that's how we've been able to identify and potentially reduce batch-to-batch variability.